Good evening, everyone. Thank you for joining us tonight in the second session. We have almost 4,000 attendees. We're excited to talk about NK and different CAR T cells. We have with us today Dr. Rafet Basar from MD Anderson. Dr. Rafet.
Hello.
Thank you for joining us tonight. We're eager to listen to your talk. It's a very exciting time to add on to the arsenal of the different cars. Dr. Rafet Basar is an assistant professor in the Department of Stem Cell Transplantation and Cellular Therapy at the University of Texas MD Anderson Cancer Center. He has developed expertise in multiple immune cell subtypes, including NK cells, T cells, and B cells, and is leading multiple exciting translational projects simultaneously. Dr. Basar, thank you for joining us tonight. We can't wait to hear your talk. The mic is all yours. Go ahead.
Okay, thank you so much, and thank you for inviting me. I'm select the forums like this beautiful. I'm select the Congress. So again, I'm select good morning, everyone, and thank you for joining our select to discuss. As he mentioned, it's like I'm assistant professor in the Department of Stem Cell Transplantation in MD Anderson. And also like to thank the organizer for inviting me to discuss chimeric antigen receptor in the application of immunotherapy. So here is my disclosure. We have selected recently the financial conflict with the Takeda Pharmaceutical Company for CAR NK cell technology. So just to basically select the mention for with the chimeric antigen receptor. Chimeric antigen receptor is the recombinant molecule consisting of the three parts. The first part, as you saw here, is the single chain variable domain, which is the antigen specific and from monoclonal antibody. And the second, they have the transmembrane domain, which can be, which I'm going to mention about like the importance of this domain. And the last is the signal transduction domain of T cell receptor, which is usually they use CD3 zeta. So when CAR T cell recognize specific antigen, the cell is activated via the intracellular signal transduction domain and the exertant target cell toxicity. So just basically, I want to select a little mention about how T cells are working, and when you put the chimeric antigen receptor on the surface of T cells, how they are working. Normally, T cells they interact with the antigen specific antigen presenting cells, such as dendritic cells, to be activated via the T cell receptor. But to augment them select this activity, they need to have them select some other select the costimulatory domain. When I talk about them select the CAR generation, why it's so important, we're gonna see them select over there. And after this engagement, T cell they start to activate and they kill the target. And this all is MSC dependent. Is important to know this. But when you come to select the CAR T cells can recognize with the SCFP part of like these CAR T cells via antigen recognition domain and is not MSC dependent. When CAR T cell recognize a specific antigen, the cells are activated via the intracellular signal domain and exert target cell toxicity. So here you can see like the different generation of like the CAR T cells from the first generation. Then now just like for CAR NK cell that I'm gonna mention the fourth generation armored CAR T cells. So as you know, just like they refine them like gradually, and the first was just the first generation just with the CD3 zeta without costimulatory domain and with the ACFE. So this first generation showed limited expansion and anti-tumor efficacy because the CAR T cell expansion in vivo was just dependent on interleukin production. But when you select the thing, as I mentioned, select in previous slide, for T cell in vivo T cell activation is caused by interaction between antigen presenting cells via T cell receptor, and they need to augment them select the T toxicity. They need them select several select the costimulatory receptors such as CD28, 41BB, which is you can see select for second generation select the CAR that develop select right after that, which select to increase the persistent anti-tumor activity, they put like another costimulatory molecule, either like 4-1BB and CD28. And then there is like in two different like the drug in 2017 when they accepted one of them with the 28 for like the higher antitoxic activity, and another like the for higher in vivo positive for 4-1BB. To further argument them selected this anti-tumor activity in persistent, we have them select the third generation, and then as I'm gonna mention them select for current cases, we have them select the fourth generation that armored with the cytokines such as IL-12, IL-15, that they can improve the proliferation and the in vivo persistence of these CAR cells. So you can see here briefly how I'm select the patient CAR T cell generated, and then I'm going to select the patient. It's quite select the complex, and the time I'm select the consumer I'm select the process. So then patient I'm select are collected via leukapheresis, and then subsequently transport in a manufacturing facility for ex vivo genetic modification. First, this leukapheresis material undergoes enrichment for target T cells with the positive selection with CD4 and CD8. Then T cell undergo activation and the viral transduction, as you see here, with the chimeric antigen receptor target against the specific tumor antigen such as CD19, BCMA. And then these transduced cells further culture expanded, formulated, and cryopreserved. And then final formulate this CAR product is then shipped back after I'm selected bridging chemotherapy, and then administered to patient via infusion in single dose. So as I say, just like in the previous email, the reason selected in the United States now, we have them selected in FDA approved them selected three different products. One of them is Yescarta and the Chimera. Yescarta and the Chimera is they approved by FDA in 2017. Yescarta for diffuse B-cell lymphoma, and Chimera is for acute lymphoblastic leukemia. But later on in 2018. They accepted, approved for the diffuse large B-cell lymphoma. And recently also like for mantle cell lymphoma, again from the same company, Tecartus approved for mantle cell lymphoma. Actually, I'm select one of my slide is missing. So, but in that I'm select the slide we just like with the Chimera and the Yescarta. There was like the clinical select trials. Dr. Benjamin I'm select to mention about this. I'm select the clinical results with the Yescarta and the Chimera with the diffuse large B-cell lymphoma. There was like less response, but with the both of them with the acute lymphoblastic leukemia. Both they have select around overall select response around 80-85 with the like 50-60% of the complete response. And the Tecartus recently it was conducted by MD Anderson by Dr. Wang also. It was a mantle cell lymphoma. They got 85 overall response, and 59 is 59% of complete response. But like for all of them, again, there was like really missed side effects with CRS and neurotoxicity. So this figure summarizes the limitation of the achieving durable selective remission with the chimeric antigen receptor CAR T-cell therapy. Quickly, I'm going to mention about this. First of all, CAR T-cell failures have several causes for some patients. And this CAR cell product cannot be successfully manufactured, or to generate the CAR T cell do not expand sufficiently. This either can be manufactured in vitro or after administration in vivo. And in other patients, also the problem of limited persistence in vivo is potential mechanism of underlying selected disease relapse. And the second that is really like the common also this is the antigen modulation. So depicted by the loss of downregulation. See some like this in the clinical trial. We have seen them select with CD19, CD22. Select on malignant B cells. These select when they downregulate these, they enable antigen escape as a mechanism of resistance to CAR T cell therapy, which can also be a problem with the non-B cell malignancies as well. And the third, like as I mentioned in previous slide, also the characteristic toxicity of CAR T cell therapy, primarily like the severe, like the cytokine release syndrome, almost like the around the 50-60% of patients they experience the neurotoxicity or severe like the cytokine release syndrome. And these like the side effects they can be fatal, but this does like abolishing the potential of therapeutic benefit in a small proportion of patients. So, and then furthermore, data on the effect of the therapeutic intervention of the CRS and the durability of CAR remission is remain unknown. And finally, unmet needs include some disease contexts that are a focus for ongoing research efforts to optimize the clinical utility of CAR T cell therapies. For example, although CD19 CAR T cell can provide substantial benefit for adult lymphoma, the complete remission rates are lower than those achieved in patients with the leukemia. And additionally, the outcomes of this might be like because of the they have much more like the complex microenvironment. And additionally, the outcomes of CAR T cell therapy in pediatric patients with the lymphoma, in patients with the central nervous CNS disease, remain in array of ongoing investigation. And notably, such therapies currently have limited efficacy in patients with solid tumors, and approaches to optimize response are being explored. So as I mentioned, I'm like the autolog, like now what's available. Dr. Benjamin also mentioned about like the effort that they're trying to like move the allogeneic CAR product. CAR T cell production, this complex manufacturing process is compounded by time-sensitive logistical matrix with many stakeholders and potential of failure. And these and other factors are challenged to be overcome with autologous like the CAR T cell therapy. As you see here, we need to move with some like for CAR T cell or other like the immune subset, we need to move to allogeneic. For allogeneic can be like from PBMC, umbilical cord blood, embryonic stem cell, or immunopluripotent stem cells. We have like off-the-shelf product. This is going to eliminate the manufacturing time and allows true point of care administration, and then can scale much larger numbers of like the product. So as again, as I mentioned, in the previous the presentation, we have like the possible possible target that we can move with the CAR T cells for allogeneic product, but what we can like the target first problem with the like HLA class 1, if is recognized by like body the T cells, they can be like reject. And the second, if you have endogenous TCR, they can attack somatic cells and they can cause the GVHD. To overcome this, like the recently last couple of years, like the CRISPR-Cas9 gene editing technology actually is not just CRISPR-Cas9, but with the TALEN, with zinc finger, they become like really popular. With that, as you see here, you can remove like the endogenous HLA class 1 and 2, and then with that, just like the patient T cell, they're not gonna recognize, and they're gonna have like long-term survival. And then when you remove the endogenous TCR from the T cell that we're gonna infuse, then you're gonna avoid from the somatic cell attack that probably can cause them like the GVHD. So there is the clinical trial which Dr. Benjamin mentioned about that with this, like the overall response in the beginning was good, but when you look at them, like the overall response was not like really good, but working on it, it's like it's still research on that is ongoing. So today actually is like since we have like all these challenges about them, like the CAR T cells, and the researchers start to look like other possible immune cells that we can use for CAR like the cargo, and then we can use as effector cells. So in our lab with Dr. Rezvani, she started to work with the NK cells like over 10 years. So today I'm gonna mostly I'm gonna talk about like the NK cells. Just a brief introduction for NK cells for those who are not familiar with the NK cells. NK cells are members of the innate immune system, which can be defined as CD56 positive and CD3 negative, and they differentiate in the bone marrow and possess no need for antigen priming. They are primarily like found in the blood, and the key advantage that Encasa provide is that they have little to no risk of the GVHD. Actually, there is a lot of like the studies is going on about that Encasa can be used for GVHD actually. And this another characteristic of Encasa is that the recognition takes place through the complex array receptors. So when you compare CAR Encasas with the CAR T cells, they have like the number of like the advantage of CAR Encasas, that we can produce allogenic product is the off the shelf with the possibility of procuring more than 100 doses, which I'm going to show you like in the next slides, from only one cord blood unit, which impact the cost of production of treatment. When you think the price of the CAR T cells for each single dose, so it's really like the big achievement with the Encasas. So combining these advantages with the low likelihood of GVHD and the other side effects such as CRS, which is common in CAR T cell therapy. And in addition, CAR NK receptor provide a functional advantage as NK cell can bind ligands expressed by tumor cells such as MICA, MICP, and LPP. But when you think about the CAR T cell, they're gonna kill them their target just with the CAR mediated killing, but CAR NK cells, they can kill by their receptor, natural ligands, and the receptors, and by CAR as well. So CAR T cells, I show you like the same slide. Actually, there is no really like the big difference between production of the CAR NK cells. Again, number of researchers they're using them like the different source of NK cells. They're using PBMC, NK92, umbilical cord blood, embryonic stem cells, and iPSC. So from these, they sorted the NK cells with negative, positive selection. Then these NK cells, I'm going to mention about them, like the aurums, like the protocol about the expansion. They activated or expanded this as then they transduced with the chimeric antigen receptor with the viral vector, which is we're using retrovirus. And then they're giving them like the more activation and more expansion. And then after that, they infuse the patient. Again, I'm like the single dose. So in our lab, in MD Anderson, we have huge cord blood banks like the facility. For that reason, just we start to move with the cord blood. The advantage of these readily available from global like cord blood bank, and these lymphocyte composition include 15-30% of the NK cells. And the more important, these NK cells, they have high proliferative potential transcriptomic profile. They have, of course, some disadvantage. They're numerically few and therefore requires ex vivo expansion. And there is variability between products. Some of NK cells from some cord blood, they are doing better than the others. So, as I mentioned before, NK cells are a good alternative for immunotherapy with many advantages in regards to side effect and cost effective and availability as one of the shelf product derived from cord blood. To take these like the step further, our team developed an armored NK cells, which is like the 4th generation CAR with enhanced anti-tumor activity. So we decided to transduce our NK cells with the chimeric antigen receptor. In this schematic, you can see the design of our construct, which was generously provided by Pietro Dotti from Baylor College of Medicine. So they compromise suicide gene just in case of toxicity, along with costimulatory domain with CD28 and CD3 zeta, and to improve proliferation and anti-tumor activity, we incorporate with IL-15. So this is my colleague Emily Liu. She transfused them selected NK cells with the chimeric antigen receptor, then tested their in vivo persistence and anti-tumor activity after infusion in NSG mice. And then the mice, they engrafted with the Raji, which is really a highly aggressive B-cell tumor model. As you can see in the first like the row, just like in 2 weeks, non-engineered NK cells and the tumor alone, they succumb with the tumor. when you have like the CD19 CAR without IL-15, they do get control some, but like after a week or 2 weeks, they again like this come with the regime, and then finally they died because of like the tumor toxicity. When you come with the CAR-19, they incorporate with the IL-15, they control like tumor with much significantly better when you compare with the other group. And even I didn't show here, but it's like even day 70, when you look at them, select the peripheral circulation, you can see them select NK cell population that we infuse, which is really important. Usually, select is the one of the limitation of NK cells that they don't select the persist as good as T cells in vivo, which means like with the IL-15 that it seems just that we overcome from this limitation. So using this method, we went to FDA and developed a GMP production protocol for expansion and transduction of current cell from cord blood. We get our frozen cord blood, thaw them, cycle them, deplete PET and monocytes, and then the cells are expanded with the K562 based feeder cells, which we generate in our lab, which is they express IL-21. And besides, with some receptor, they can improve the proliferation and the anti-tumor activity of NK cells, 41BB and CD48. And then in the culture, we support with the IL-2. On day 6, we transduce the cells with retrovirus supernatant, and then we do one more round of ex vivo expansion. So at the end of 2 weeks, we have transduced NK cells ready for the patient infusion. Before I'm select the infusion, of course, we're checking them select the phenotype of NK cell to make sure there is no any contamination, and we have the poor population CD56 positive, CD3 negative, and CD16 positive. So this table summarize the data on the expansion and the transduction efficiency of 5 different cord blood. I think just like the deal was gonna come. I'm select after I'm select the slides, but it just like the close. I'm selected the first 2 cord bloods. But we got them selected from the 5 different cord bloods. From each cord, we usually get around 20 to 50 million NK cells. And at the end of 2 weeks expansion, we can expand NK cells more than 2,000-fold with the excellent transduction efficiency, which is like the average 66%. And the absolute number, like around 10 billion current cases, we can achieve after 2 weeks. So this means that from one cord blood unit, we can potentially generate roughly around 100 doses of current cases at 1 million per kilogram. So with that, just like these, I'm selected by Dr. Katie Rezvani, MD PhD from MD Anderson Cancer Center, and we started a trial with the current cancer therapy in patients with relapsed and refractory B-cell lymphoid malignancies. This slide summarize the schema. We picked the cord blood units with the match 4 out of 6 HLA molecules. In the first terms, like because there was not necessary for in cases, but FDA is asking in the beginning, so that's why we start with that. But the last 2 patients I'm gonna mention, we use them like the HLA, without looking them like the HLA match, we infuse the cells, and the patient comes, and then again we told the course select the NK cell, expand for 2 weeks, and then patient received lymphodepleting like they are doing with the CAR T cell with the cyclophosphamide and fludarabine. And then because of this was like the first clinical trial, we start with very low doses, like with 100,000 per kilogram. Then we move to up to like 10 million per kilogram. So, so this
2 minutes, Dr. Basar.
Okay. So with this is like our clinical summarize. Totally, we have like the 11 patients, and this is like 5 of them is CLL, 6 of them like the diffuse large B-cell lymphoma. The overall we got them like the 83 response, and the 7 of them like we have them like the complete response. And when you look at them like the last two patients also it's HLA unmatched, and we didn't see any toxicity. Is the most important. Without any with any patient, we didn't see any neurotoxicity and the CRS. So this is the overall survival and the progression of selective free survival. As you see here, just like the overall survival is around like the 83%, with the progression of selective free survival is like around 79. So this is the one patient that with the Richter syndrome, they got them to complete remission. This is like the 3 weeks ago before like CAR NK cell infusion. After we infuse CAR NK cells, after 30 days after PET scan, as you see here, they clear all the tumor in the body. So, and then when you look at them, select the mass cytometry from the same patient, as you see here, this is the B cell, select the tumor burden. After 6 days, they remove all select the tumor burden, and they're really good select the NK cell engraftment. And this is another patient again. Select with the high select the tumor burden. After this is the pre-admission, and after you infuse the CAR NK cells, it says again select all select the tumor gone. There was like only just little bit select in the armpit. And when you biopsy these and then look at them select the composition of cells, they're all coming from the donor NK cells actually, which means just that they can traffic also tumor site. So, and then like one concern that we've been seeing like with the CAR T cells, we look at the severe the cytokine release syndrome. There is no any upregulation or cytokine release syndrome related cytokine. So just like the 2 slides is like more, as you know, just like since we haven't like the good results with these, just we're trying to improve that. As you know, just like there isn't really a good checkpoint for T cell, but there's no really well known just like the checkpoint for NK cells. They recently by Nick Huntington, they defined them like the CISH, which is like the breakpoint for JAK-STAT pathway. And we decided and the knockout select this gene with the CRISPR-Cas9 to improve select the NK cell activity. And as you say here, just like after you knockout, you can see just like how select the improved CAR NK cell functionality with no evidence of tumor with any organs that we check in pathology. So, as a conclusion, the CAR T cell revolutionized the treatment of the B cell malignancies, and the NK CAR is really good, a good alternative for CAR therapy, especially after it is the first human clinical trial with the CD19 IL-15. So, with that, as I say, just like the CAR T cell now in the guide, but in the future, NK cells and iNKT cells, and the recent CAR macrophage on the way. I think just soon there are going to be clinical trial about this. With this, I want to thank, and this is my lab, my mentor, Dr. Rezvani, and my colleagues, May Dahar and the other, and then all stem cell transplantation. Thank you.